I actually really like the way these talks have been ordered, because I feel like everything's obviously pretty much everything's been related to microscopy in some way, but we've built from really small things all the way up to now something very large. So what I'd like to talk to you about today is a group of techniques known as tissue clearing techniques. And the way I want to do this is I want this talk to be more like a lecture in a classroom, let's say. I'm going to talk a little bit about research right at the very end, just to show you an application. But what I really want to do is teach you about these techniques, how they work, in the hopes that you might be able to take some of this and apply it to your own research. So as far as a quick outline goes, what I want to talk to you about first is how light scatters when it comes into contact with matter, and why that is an issue when we're dealing with fluorescence microscopy, and how this limits the size of samples that we can look at under the microscope. After we've explained a little bit of very basic physics behind that, we're going to talk about the actual tissue clearing process, which is essentially a lot of chemistry. And I'm going to talk you through the different stages of a tissue clearing experiment and give you an idea of what each of those different steps is doing on somewhat of a molecular basis. Then the next thing I want to talk about is objectives and microscopes that we use for imaging these types of clear tissues. Microscopes historically have been designed for imaging really thin tissue sections and small slices of biological samples. And so this idea of putting something that's millimeters thick or even centimeters thick under a microscope is just, when these techniques came around, it created a lot of headaches for microscope manufacturers, let's put it that way. I'll briefly touch on data handling, just to give you kind of a warning that the bigger an object you're imaging, the more data you're going to have to deal with. And then, like I said, I just want to close with a final example of some work that we've done in collaboration with Kate Bentley's group in imaging the vasculature of the mouse retina. Great. So here's the problem. So on the left, we have a mouse brain. So this brain was, the mouse was anesthetized, the brain tissue was fixed, it was removed from the mouse, sat in fixative overnight, and this is essentially what it looks like. This particular mouse it's called a Phi1 GFP mouse. So what that means is that in a subset of neurons in this mouse brain, it's expressing a green fluorescent protein behind the Phi1 promoter. And this is one of the greatest samples to use for these tissue clearing experiments, because it's just these neurons are incredibly bright and very easy to detect. So having said that, this is one of the brightest samples out there. If you just put this under a microscope as is, so let's say a confocal microscope, which allows us to take optical sections throughout the sample. And if we start imaging at the top and try to image down into the sample, here's what happens. I'm going to play a little movie on the right. So this is, you can just see there's just a little bit of autofluorescence here. We're just right at the top of the brain. I think we're somewhere in the cortex in this region here. And what you can see is we start imaging in, we can start to see these nice bright neurons. And then all of a sudden, really quickly, everything fades to black. So even that autofluorescence that should be coming from the brain tissue everywhere, we're unable to see that anymore. We just lose all of that signal. And just to show you this a little more directly, here's a cross-section across this data set. So somewhere in the middle here, a cross-section. And what you can see is we've only gone 60 microns into that sample before we've lost pretty much all of our signal. So the objective that we're using on this particular microscope, it's a really good objective. It has a working distance of over 2 millimeters. So we should be able to go a lot deeper. But there's something that's preventing us from being able to image those deeper structures. And the reason that we're unable to image deeper than that is what's quickly referred to as light scatter. So this is something we should be all familiar with. Light scatter is the reason that the sky is blue and the sunset is red. But I want to give you sort of a more practical example here that sort of relates to what we're seeing in this biological tissue. So this is a really simple experiment that you can do at home. All you need is some sort of sample holder. So what I have here, this is just a little plastic cube. Let's open on the top. I filled it with pure water. I just took a laser pointer. But if we weren't on the computer here, we were actually in a lecture hall I'd be using today, and shined it through that cube. And so you can see at the edges here, we get a little bit of reflection. But if we look in the middle of this cube, we don't see any of the laser light. And what that means is that that laser light is traveling in a straight line directly through here. It's not scattering out towards the camera. We don't see it coming up out of the sample. All of that laser light is passing straight through. So this is what we would generally refer to as a sample that has no light scatter at all. But what I'm going to tell you right now is that's not true. There's actually lots of scatter happening here. It's just a very specific type of scatter, which I like to often refer to as homogeneous scatter or homogeneous interference. So if we take a look at this laser light, this green laser light that's coming into our cube, and we have a bunch of water molecules right at the edge of our cube, these black arrows represent that laser light coming in. That laser light is going to hit some water molecules that are lined up right at the edge there. And whenever light interacts with matter, it can scatter. And what scattering means is that light, the photons of light that are coming in here are going to be briefly absorbed by the electrons in these water molecules. And then very quickly, they're going to be spat back out, but they can come out in any direction. So the light coming out looks exactly the same. It has the same wavelength, the same amount of energy. There's no change in color or anything like that. There's no energy lost in this process. But the only difference is that it can come out in any direction. Okay? And sorry, there's one other difference as well, is that this interaction obviously takes time. So it means that if you want to think of light as a wave, that wave has been slowed down a little before it comes back out. So it's been shifted. Okay? So if we just look at these four molecules that are all in a line here, and we have light coming in from our laser beam, it hits all these four molecules at exactly the same time. Light scatters off of all of them at the exact same time. It can go out in any direction, but for the sake of this discussion, let's just look at the lateral direction. Let's just look at each one of these molecules scattering light directly straight up. And so what's going to happen here is within this cube, these water molecules are all packed in really tightly. And the water molecules are really small compared to the wavelength of light. And what that means is that any molecule of water, there's going to be another molecule of water that's exactly half a wavelength away from it. And so when these molecules that are exactly half a wavelength away from each other scatter this light, you can see this blue line here is the wave of light that's been scattered off the blue molecule, and the yellow line here is the wave of light that's been scattered off the yellow molecule. You can see these are exactly out of phase with one another. So we have the yellow one going out to the left here, the blue one's going out to the right. And what that means is that those waves of light are going to interfere with one another, and they're going to interfere destructively. So what that means is we're not going to see any light going out in this direction straight up. Likewise, this green one and red one, they'll interfere with each other, and so we don't see any light going in that direction. And it turns out that this is the case for every direction. So whether you're going up on an angle, backwards, down on an angle, this is the case in every situation except for one, and that's when the light is going straight forward. So in the case where our light is going straight forward, what we do is, instead of exciting a whole bunch of, or scattering off a whole bunch of molecules at the same time, we scatter off molecules in succession. So this green laser light that's coming in, some of it scatters off this red molecule first, then some of it scatters off the blue molecule, some of it scatters off the green molecule, and then the yellow one, all in succession. And so what happens here is a little bit of a different process. So here's our green laser light wave coming in. It interacts with this red molecule, and the initial wave keeps moving. So it moves on to the blue molecule. But remember, I told you that scattering event, there's a little bit of a pause there. So that light that's going to scatter gets absorbed for a brief moment and then released. So what that means is that when the scattered light is released, it trails half a wavelength behind the initial wave. Okay? So now we have our initial wave that's now interacting with the blue molecule here, just as our scattered light from the red molecule is being released. And this continues on. So now we have the light wave interacting with our green one when the blue one is released, and now it's interacting with the yellow one when the green scattered light is released. And what you can see here is that instead of having destructive interference in this case, we have constructive interference. So all of these waves are matching up exactly in phase with one another. And so that means that we're going to be able to see all of our light energy moving in this forward direction. So this is the only direction that this is going to happen in. In every other direction, we get this destructive interference. And that's what allows this light to move directly through this cube from one side to the other. And if you put a power meter over on this side, you would detect virtually the same amount of light energy as you see coming out of this laser pointer. Okay? And this is the case for not only pure water, but we can also mix something else into this solution and still see the same thing happen. So here what I'm going to do is I'm going to show you another cube on the other side that also has water in it, but in this case we've added just a few microliters of ethanol to it. And when we do that, we see exactly the same thing. Okay? So once again, there's no scatter. The light is passing directly through. So what's going on with biological tissue? Well, biological tissue is full of lipid and protein. And there's something else that we come in contact with most days that's also full of lipid and protein. And that's the cream that we put in coffee. So let's repeat this experiment, but instead of adding ethanol, let's add a little bit of coffee cream. And I'll tell you right now, if you put 10 microliters of coffee cream in a cube that looks exactly the same as this one, you hold them up in front of your face, you can't tell the difference. You don't know which one has the coffee cream and which one's the pure water. There's so little cream in there. Once it dissolves, you can't tell the difference between them. But if we shine our laser beam through it, now all of a sudden we can tell there's something different in that cube. So we have all kinds of light being scattered out of that cube coming back to our camera. So that means our light that's been moving forward in this direction is hitting something in this cube and shooting straight up at our camera. And it wouldn't matter where we put our camera, anywhere around this cube, we'd be able to see that light. It's being scattered in all directions. So what's special about this coffee cream that, or what's different about this coffee cream that allows this process to happen that ethanol wasn't able to do? And the major difference here is that those lipid molecules and protein complexes are really large objects that are sitting inside the solution. So we have all these water molecules that are nicely packed in tight to one another, but all of a sudden they're interrupted by these really big blobs of lipid and protein. So these can be they're obviously much larger than the wavelength of light. They can be up to 10 micrometers in size. And what happens is they disrupt that lateral destructive interference. So you can see these water molecules here. There's no other water molecule close to them to scatter that light. And by breaking these water molecules apart, that allows light scatter to happen in all directions, and instead of that deconstructive interference, reducing it in the lateral dimensions. So now, if you put a power meter over at this side of the cube, what you would find is that the energy of the laser beam over here has been reduced compared to what's coming out of the laser. And that's essentially what's happening in our microscope. So if we're trying to image fluorescence deep in that mouse brain that I showed you earlier, our laser that we're using to excite our sample, as it tries to be focused deeper and deeper into the tissue, it's losing more and more energy as that light scatters away from it. And probably more importantly is that the fluorescent molecules that we have deep inside our tissue that are giving off that fluorescence, that fluorescent light that's being emitted is scattering on its way back to our objective. And so this is why things get darker and darker as we image deeper and deeper with our microscope. So I told you that the big reason that this coffee cream is scattering is due to the size of these objects. It's not really necessarily due to the size. It has to do with the electron shell around the molecule. It has to do with the density of the molecules. But there is one measure of a substance or substances that we can use to know whether when we mix them together or not, we will see this light scatter. And that measure is something called refractive index. So if we mix together two substances that have a similar refractive index, so like water and ethanol, water has a refractive index of 1.33, ethanol I think is about 1.34, maybe 1.35. Those are so close together that we're not going to see a lot of light scatter. However, this coffee cream, these big lipid droplets and protein blobs, these have a refractive index up around 1.47. So there's a big refractive index mismatch there, and that's why we start to see this light scatter so predominant. So what this means is that the big secret to tissue clearing is trying to address the different refractive indices that we see within our tissue. And there's three major refractive indices that we have in any biological tissue. So the first is our intracellular and extracellular fluid, which is essentially water, salts, and a little bit of protein. And so this has a refractive index very close to water, just a little over 1.33 at about 1.35, 1.36. We have a lot of lipid in our cells. These are one of the largest contributors to light scatter. And so especially in subcellular organelles that have dual membrane layers, like mitochondria or Golgi. And so these have a refractive index up around 1.45 or 1.47 even. And then we also have a lot of protein in our sample, obviously. And proteins, especially structural proteins like collagen and elastin, or proteins with large hydrophobic regions, these have very high refractive indices. So these are up over 1.5, can be upwards of 1.55, 1.56. So our secret to a successful tissue clearing experiment is to somehow equilibrate all of these different refractive indices, which is going to give us some nice homogeneous interference. We're going to reestablish that destructive interference in angles other than the forward direction of our excitation light or emitted fluorescence. And in the end, we're going to get a clear tissue. So if there's some magical way to equilibrate all the refractive indices in this brain, we would be able to turn it into something like this, which is nice and clear, so much so that you can actually see these black lines of this grid through that tissue, which is almost a centimeter top to bottom here. Okay. Cool. So how do we go about doing that? There are tens, maybe at this point there might even be 100 different published tissue clearing methods. they have a whole bunch of different acronyms, and it can get really confusing when you dive into the literature. So I don't want to talk about each individual method. Obviously, that would be very time consuming, and we don't have that time. But there are a number of components that are 100% common between each of these techniques. And so if we think of the clearing experiment as a whole, there are three major components to it. So the first one is the sample preparation, and this is actually the clearing process. So this is trying to take that cloudy sample and turn it clear like that. And unfortunately, that's only about half of the experiment, sometimes even less than half. After that, you've got to somehow figure out how to image that under a microscope, and then you've got to figure out how to store that data and analyze that data. So I'm not going to spend very much time on this today, but I want to take some time to work through the sample preparation and a little bit about how to image these samples. So the sample preparation process, again, I said there's so many of these different methods out there, but all of them have three key steps to them. So most of them will have some sort of pre-treatment to them. There will be a step of delipidation, where we're trying to remove that lipid from the sample. Then there'll be a labeling step, and then there's going to be a final refractive index matching step. And just to kind of show you what that looks like as far as the samples go, here's an example of two pieces of tissue. So on the left here, we have some skeletal muscle from a mouse and a mouse brain. And we do some initial pre-treatments to that. Most of that is trying to wash out all of the blood. So you can see the skeletal muscle tissue here has gone from this pinkish color to this nice tan brown color because most of the blood has been removed from it. Then we move on to a step where we try to remove as much lipid as we possibly can. And you'll see these samples are still very opaque. They're not clear. You can't see that grid through this one. And although this one's a lot smaller, you're able to almost see the lettering behind it, but it's still not perfectly clear. But you can see it's gone to sort of this more yellowish, gelatiny-like look to it, as opposed to the firmer pinkish tissue that it started out as. Then if you're doing some immunolabeling, so if you don't have any fluorescent proteins in it, you would usually do this at this point after you've removed that lipid and made the tissue a little more porous. And then we go on to our final clearing step. And this is where we put it into a high refractive index solution. And this is where we actually can watch that tissue clear over time, and where we'll be able to see nicely through it, and we can see the text or the grid lines underneath that tissue very clearly. So what I want to do is just work through these four steps with you and just give you an idea of how these work and why each one is important. So the first step is pre-treatment. And one of the most important things about pre-treatment is making sure that all of the molecules that you're interested in are still going to be there at the end of your experiment when you start imaging. So the clearing process itself can be quite harsh. A lot of the treatments, especially the delipidation process, can remove other molecules other than lipid. So you want to make sure that your fixation protocols are really good and excellent at preserving the molecules that you want to see in the end. So anyone who's worked with fixed tissue before is familiar with aldehyde fixation. So usually we're often doing this with paraformaldehyde. And paraformaldehyde is just this sort of dual linker molecule that can grab onto proteins that are neighboring one another and just hold on to them nice and tightly and keep them in place. So paraformaldehyde works well for fixing our tissue at room temperature and keeping it in that state for fairly long amounts of time. However, it's not perfect. So if you took a sample that had yellow fluorescent protein in it, fixed it in paraformaldehyde, and then tried to do a tissue clearing process at a really high temperature, so up at 70 degrees Celsius, what you'll see is that that YFP molecule is going to denature. It's going to fall apart at that high temperature. and when you go to image your sample, there's not going to be anything left. So sometimes one option here is to use different types of fixatives. So glutaraldehyde is a good one, although it creates a lot of autofluorescence. So what's been shown in this paper is the use of this epoxide molecule, which instead of having 2 arms like paraformaldehyde, it actually has 3 arms. They're a little bit longer, a little bit more flexible. And so it can create a lot of intra and intermolecular bonds and really hold that molecule nice and stably. And what you see is when you use that epoxide along with paraformaldehyde, you can actually preserve the structure of that YFP molecule or TD tomato even after you go through these really harsh treatments. Another thing to think about is if you're interested in looking at mRNA instead of protein, paraformaldehyde does not do a good job of fixing genetic material in place. So here we can once again use this epoxide. This reagent's called SHIELD. This is out of the Chung lab at MIT. Or you can use another chemical that's more traditionally been used called EDC. And you can see that these do a much better job of preserving genetic material in comparison to paraformaldehyde. Another method that's very popular in tissue clearing is to embed the entire sample in a hydrogel. So this is usually an acrylamide gel. We allow that gel to impregnate the tissue, and then we cross-link our protein onto the gel. This is a way to specifically retain protein. The lipid molecules will not cross-link to that hydrogel, and this allows our delipidation process to work better. We can be really harsh when we're trying to pull that lipid out because it's not attached to that hydrogel, and our protein will stay behind under those harsh conditions. Okay. So this idea of fixation is really important during your pre-treatment. The other thing that you have to think about in pre-treatment is this is essentially what every tissue clearing protocol is going to do. So we're going to remove that intracellular, extracellular fluid, which is usually water. We're going to replace it with a high refractive index solution. We're going to try and get rid of all of those lipids that we can. And this solution that we use to replace the intracellular and extracellular fluid, it's going to have a nice high refractive index, and we're going to try and match the protein that's been left behind. So this is the basis of every tissue clearing experiment. Remove that intracellular fluid, remove the lipids, and replace that intracellular fluid with something that's matched to the protein. So what you can hopefully appreciate from these 3 steps is that in no way do they address autofluorescence coming from your tissue, and in no way do they take care of any pigments that you might have in your tissue. So this is something that you have to take care of in this pre-treatment step as well. So if you have a tissue that's really autofluorescent, one way you can do that is to build one of these units. It's basically an LED array of high-powered white LED lights, and you just sit your tissue under it for a few hours or overnight, and that bleaches out a lot of this autofluorescence. Of course, if you have fluorescent proteins in your sample, it's going to bleach those as well. So be careful in your use of this. If your tissue has a lot of pigment in it, so this is usually the key problems here are heme groups. So if you have very blood-rich tissues that have a lot of hemoglobin in them, or if you have muscle tissue that has myoglobin in it, you need to get those out. Another example is melanin. So if you're dealing with skin tissue, you often have melanin pigment. You need to get those out during this pre-treatment phase. So this is something we call decolorization or bleaching. And this is often done with hydrogen peroxide, which is the best for getting rid of melanin, really dark pigments. And there's also another family of molecules called amino alcohols, which can be used to get rid of the heme groups in your sample. And you can see we can take these really dark tissues like kidney and really get rid of all of that pigment so that our light can pass through them better. And even tissues that you wouldn't traditionally think of as being pigmented, like brain tissue. This really comes into play with human brain tissue. It's a lot more pigmented than animal. But you need to get rid of that pigment in those tissues as well. And this all happens during the pre-treatment phase. So now we've got a sample where we've gotten rid of the autofluorescence, we've gotten rid of the pigment, we've got the molecules that we're interested are really nicely fixed in place. So our next step is to try and get rid of all the lipid. And this is where we really start to see two branches of tissue clearing. So there's a whole group of tissue clearing methods that are called aqueous-based techniques, and that all that means is that there's going to be water present in your sample throughout the whole clearing process. And then we have another bunch of techniques that we call solvent-based techniques, which one of the first steps during that pre-treatment phase is going to be to dehydrate your sample and get rid of all of the water. And then all the subsequent steps are going to be performed in the absence of water using a number of different solvents. So as far as this lipid removal step goes, our aqueous-based techniques use detergents. So these are detergents that you're likely familiar with. So we can use SDS, Triton X100, CHAPS. Whereas on the solvent side, we use two solvents in particular, tetrahydrofluorine and dichloromethane. So this step, you have to be a little careful with it. This lipid removal process can really distort your tissue. And so if we take a look here, this is a mouse brain on the left. And if we, in general, our solvent clearing techniques shrink our tissue, and our aqueous techniques expand our tissue. Sometimes this is very advantageous. If you have a really large sample, you want to shrink it so that it's going to fit in your microscope and be easier to image. If you have a sample that's really difficult to clear, sometimes expanding that tissue helps to clarify it a little bit more. But you do have to be careful. So down here is just an example of, this is a mouse embryo that's been cleared. In this case, the delipidation was done with THF, whereas down here it was done with methanol and DCM. And what you can see is that this embryo becomes really distorted. These spaces grow dramatically. The rest of the tissue shrinks. When you use THF, it's a really fast, really quick delipidation process. Whereas if you do it a little bit slower and more gently with methanol and DCM, you much better preserve that tissue overall. And another thing that's interesting is that solvents and detergents, lipids are not homogeneous. They're very heterogeneous. And there's some lipids that are removed better with detergents, and there's others that are removed better with solvents. And so when you have something like adipose tissue, it actually turns out that using both a solvent and a detergent can allow you to clear that tissue a lot better. So this is just an example of adipose tissue here that used this technique called AdipoClear. You can see it does a really good job of getting a lot of that lipid out of the sample. You can see the black background much better here. Whereas if you use a solvent-based protocol alone without any detergent, it doesn't do as good of a job. So if you have really, really lipid-rich tissues, a combination of solvents and detergents can work really well for clearing out that lipid. Okay. So now what we've done, we've removed all that lipid from our sample, which means our sample is a little more porous. And if we have a tissue that we didn't have any fluorescent proteins in to begin with, we need to get some sort of fluorescent label into our cell. And so this is done with immunolabeling, of course, which is something we're all familiar with when we do fluorescence microscopy. But the difficulty here is that if you've ever done fluorescence immunolabeling on, say, a monolayer of cells, you've probably done some sort of optimization. But it really goes to the next level when it comes to optimizing your immunolabeling for working with these really thick tissues. So usually the first time anyone attempts one of these experiments, this is what they see on the right here. It's just all kinds of non-specific staining that's all localized to the outside of the sample. the antibody just doesn't penetrate to the inside. It doesn't find its actual target. And so there's a large number of reasons why this may happen. You might just have a poor antibody from your supplier. It might be cross-reacting with nonspecific epitopes. You may need to play around with your primary concentration. You may need to play around with your secondary concentration. You definitely have to give this a really long time. It usually takes 5 days for a big sample, like an entire mouse brain, for that antibody to penetrate all the way through it. You may have to work on temperature. Your antibody itself, it's a protein, so if you're sitting it out at room temperature or at a higher temperature, it might denature over time, so you might need to do this at a colder temperature. Your fixation can come back to affect how well that antibody can find its epitope. And if you're working with one of these techniques where you have hydrogel in there, if your hydrogel is too dense, there aren't enough pores in that hydrogel for the antibody to squeeze through, and it gets stuck on the outside as well. So this can become the greatest barrier to tissue clearing, is going through this optimization, and it can be very expensive. So the one way to go about this is to don't ever start with an entire brain. Cut it up into some thinner sections, maybe even a millimeter at most, and work on finding a protocol that gives you some nice deep penetration of your antibody. Once you get that worked out, there's a number of techniques available now to help speed that antibody moving into your samples so that you don't necessarily need to wait 5 days. And most of these revolve around using an electric field to pull that antibody through. Another way to do it is to just use a different type of antibody. So human antibodies are very large. Same with rabbit and goat antibodies, which is what we're often using. However, camels and sharks have these really different antibody structures where this variable domain is pretty small, and you can actually chop these off. And once you know the peptide sequence of these, you can actually just create them in bacteria. There's no need to do a traditional inoculation of an animal. And when you have these really, really small antibodies, you can perfuse them through the vasculature and just get this amazing labeling. So this is out of Ali Ertuğ's group in Germany, where he's perfused and then cleared an entire, I think this is actually a rat, it's not even a mouse. And you can see you can get this great labeling of the nervous system all the way through. And Jeff Lichtman's group here at Harvard, he's done a good job of showing how much faster these antibodies penetrate. So this is after 48 hours at 4 degrees Celsius. You can see the yellow color is the antibody. The green color is a YFP that's already in the tissue. You can see with these nanobodies, you can penetrate a good ways in, over 100 microns, whereas if you're using a traditional antibody, you're barely getting through the surface in that amount of time. So unfortunately, these aren't widely available at the moment, but they're slowly becoming more common in use. So these can really help in immunolabeling of cleared tissue. And then finally, once you've got your sample all labeled, the last thing you need to do is match the refractive index of that protein that's left behind. And we can do this either with those aqueous-based techniques or with the solvent-based techniques. And there's a whole bunch of different solutions that are used for this. There's lots of them available. The most important aspect is to get this refractive index up over 1.5. So if you're below that, you're not going to get an adequate amount of clearing. Okay, so that's the process for clearing tissue. We've got a nice cleared tissue, and now we want to go ahead and image it. And this is where things get difficult, because traditionally microscopes were built to image these thin samples on a microscope slide, not a big, thick, multi-millimeter or centimeter-sized piece of tissue. So how do we go about selecting a microscope or objective that can actually image something this big? So the key that we need is we need a lot of working distance. So we need an objective that can image really deep into our tissue, but we also want to retain a high amount of resolution, especially in that Z dimension, in the axial dimension, because we're trying to do 3D imaging here. And traditionally, working distance and resolution, so resolution is measured by the numerical aperture of objective. Magnification doesn't really matter here. It's this numerical aperture that determines the resolution of your objective. These have been inversely proportional to one another. So here's an example of a lower magnification, or sorry, a lower numerical aperture objective is 0.57, and a really high resolution objective, a 1.4 numerical aperture. And you can see that this one has a very, very short working distance. You're only going to go a little over 100 microns into your sample. Whereas this one could image a really thick sample up to a centimeter in size, but it's going to be at a very low resolution. So how do we create an objective that has both a long working distance and a high resolution? So if you take a look at our objective, what determines the resolution is the angle of this cone of light that goes into the objective. So this is what I mean by NA, or numerical aperture, is how large the angle from this midline here to this peripheral ray entering our objective is. So objectives with long working distances have a low numerical aperture, whereas objectives with a short working distance have a large numerical aperture. Okay? So you can see this is a very small angle here, where this is a larger angle here. So how do we actually make a high NA large working distance objective? Well, the answer to that is, in this situation here, what I've drawn here, these both have the same diameter front lens. So that means there's the same width of glass in both of these that's accepting light rays. But if we take a look at these two objectives here, you can see they have virtually the same numerical aperture. So they're both around 1.5 or 0.5, but the working distance is much different. So this one has a 3.6 millimeter, whereas this one has 10.6. And the difference between these two objectives is the size of the front lens. So you can see the front lens is much larger in this one than it is over here. And so if we overlay that over each other, you can see, although the numerical aperture is exactly the same, these two lines are on the exact same angle. Because this front lens is wider, we're able to image deeper into our sample. So microscopy manufacturers have now come out with a number of objectives that allow us to have these big front lenses in them and really image deep into our tissue. So there's some tissue clearing specific objectives that you can see they have these nice correction collars, so you can adjust them to whatever refractive index you have. They have really long working distances. They're really high resolution. They're all mostly 0.9 numerical aperture or higher. And these objectives we primarily use on confocal or two-photon microscopes, whereas we still have a whole bunch of traditional air objectives that have really long working distances. And although these would traditionally have very poor Z resolution, if we use them in conjunction with a microscope called a light sheet microscope, we can actually overcome that limitation in Z resolution. So here's an example of a number of different types of microscopes that we can use for imaging these types of tissues. So, of course, we need a microscope that can perform optical sectioning, because these samples are really thick, and we need to be able to just look at a very thin plane within that sample at each time. We don't want to look all the way through it. And so each of these will do that. They're all optical sectioning microscopes, but some are better than others for different applications. So a spinning disk is nice because it's really fast, but it's still pretty limited in how deep it can image. So these aren't going to image much more than 100 microns into a sample. A point scanning confocal is nice. It can image really deep. It gives you really nice subcellular resolution, but it's going to be really, really slow. So you're not going to want to do large areas of a piece of tissue. You can go really deep into that tissue, but to do the whole tissue is going to take a really long time to do it. A two-photon microscope is excellent if you can't clear your sample very well. but it doesn't work very well for multicolor imaging. It's tough to do more than 2 colors. It's also going to be very slow. And then these light sheet microscopes, these are what we're primarily using for the largest types of samples. And they're nice because they give us a pretty high resolution. You can potentially do subcellular imaging. And they're also really fast. But once you get to this point of using these really, really large samples, you get stuck with a lot of data. So this is just a quick calculation. If you're using a 20x objective and trying to image an entire mouse brain, on a confocal microscope, you'd have over 4 million Z-planes. It would take you over 40 days. And the nice thing is we can do this a lot faster on a light sheet, but you're going to create a whole bunch of data. So it turns out to be over 30 terabytes of data per color channel. So we usually don't do that. What we'll do is we'll use a light sheet microscope with a 5x objective, which allows us to cover this tissue with many fewer images, but it still ends up being pretty big. So for a 2-color entire mouse brain, you were looking at almost a terabyte of data, but it only takes you about 3 hours to acquire that. So in the last couple minutes here, I just want to give you one quick example of what you can do with this type of imaging, with this tissue clearing. And I want to give you this example of looking at the vasculature within the retina of a mouse. So traditionally, the way imaging of a mouse retina has been done is you dissect the retina out of a fixed mouse eyeball, and you make these 4 incisions so that it gets spread out kind of like flower petals onto a microscope slide. You put a cover slip over top of that, and then you image it under a confocal microscope. And if you do that, you come up with these really beautiful images, but it's very difficult to appreciate how these different areas connect and what the actual spherical or semi-spherical initial structure of that sample was. And so Kate Bentley came to me a few years ago, and she was having this issue where she's really interested in the vasculature within the retina and modeling it computationally, but she has difficulty making these connections across these big empty spaces. So what we did was we devised a way to image these mouse eyes intact in their native conformation by using tissue clearing and a light sheet microscope. And essentially, the process is you just dissect it out, you suspend it in an agarose block, and you put it in front of the microscope for imaging. So this is what one of these samples looks like before you do your tissue clearing on it. So you can see it's got a lot of pigment in it. It's not clear at all. There's no way to see through it. If we go through a tissue clearing protocol called PACT, we also did this with solvent-based process as well called iDisco. You can clear this eyeball up quite nicely, where you can see these black lines transparently through the eyeball. And if we put that under the microscope, we can take these beautiful images of the vasculature throughout the entire eyeball. We can look at the iris or the retina, look at the optic nerve, and we can take a look in 3D here as well, where we see the nuclei in magenta and all the vasculature in green. So this was nice. And so the next thing we wanted to do was just see how much better this actually is than the confocal microscope. So what we did was we took the retina, imaged it on the light sheet, and then after we did that imaging, we took it out of this agarose block, did the traditional flat mount, imaged it on a confocal, and then compared the exact same vessels. So we found the same vessels and compared them. And what you can see is under the light sheet, we get this beautiful, nice round vessel, whereas under the confocal, it turns out that we see this very smushed, flattened image. And what this means is that any sort of quantitation that you do on the volume of these vessels is entirely wrong. And this has to do a bit with the objectives and the way they're imaged. But we can see that not only does the light sheet and tissue clearing allow us to image that entire structure in its native form, it also allows us to get a much more accurate image of the vessel itself. So just to apply this to actual biology, we turn to this model called Oxygen-Induced Retinopathy. And what this is if you take mouse pups and from day 7 to day 12, you keep them in a high level of oxygen, this actually inhibits the development of vessels in their eye. And then from day 12 to day 2017, you put them back in normal room air. And then what was thought to happen was that you had this uncontrolled vessel development where you just got these big blobs and tufts created, which were just thought to be a big pile of cells. And it turns out that if you image these under a confocal, they do just look like a big blob. But with our tissue clearing method and our light sheet imaging, it turns out that these are actually still vessel structures, but they're just piled up in this little curved pyramid. And so this was an interesting insight that no one realized that these structures were actually still vessels. They thought it was just sort of an uncontrolled pile of cells. Not only were we able to see these spirals, but we also saw these interesting figure of 8 knots as well. So this is just one example of how tissue clearing and light sheet microscopes are able to enable some new biology and challenge previously conceived notions just by doing a better job of imaging that spatial environment. So with that, there's just a couple of people that I'd like to thank. So as far as the tissue clearing and light scatter project, this started ages ago when I first came to Harvard with a couple of postdocs, Joe Bergen and Yo Isogay, who now have their own labs, and a graduate student, Will, who's now a postdoc at MIT. And they were the ones who originally brought this challenge of imaging these big tissues to me. And there's been a long time collaboration with Jeff Lichtman talking about the physics behind this and how these techniques actually work. The Retinal Vascular Imaging Project was done in collaboration with Kate Bentley, who used to be at Beth Israel here in Boston. She's now moved to the Crick Institute in London, and a postdoc in her group, two postdocs in her group, Claudia and Kyle. None of this would be possible without some commercial partners who have helped us along the way. So Zeiss, Logos Biosciences, Life Canvas, Thermo, and Arebus. And then, of course, we've had funding from NIH and an internal funding body known as Harvard Catalyst. So thanks for listening. And I'd be happy to take any questions that you